Welcome back. In Section 1, we established the foundational concepts: what a clinical trial is, why they exist, and how they fit into the drug development lifecycle. Now, in Section 2, we go deeper into the clinical phases themselves. We'll walk through Phase 1, 2, 3, and 4, what each phase is designed to achieve, who participates, and what success looks like at each stage. Let's get started. Here is our roadmap for Section 2. 4 lessons, each approximately 5 minutes, covering one phase of clinical development. Lesson 2.1 covers Phase 1, first in human safety studies. Lesson 2.2 covers Phase 2, where we test for proof of concept in patients. Lesson 2.3 covers Phase 3, the pivotal definitive trial. And Lesson 2.4 covers Phase 4, what happens after approval. By the end of this section, you'll see clearly how the phases build on each other, from a handful of volunteers in a Phase 1 unit all the way to millions of patients in post-marketing surveillance. Let's begin with Lesson 2.1. Lesson 2.1 covers Phase 1, the first time an investigational drug is given to a human being. This is one of the most carefully monitored moments in all of clinical research, and understanding how it works gives you the foundation for everything that follows. Phase 1 is the first time an investigational compound is administered to a human being. And critically, the primary goal is not to show that the drug works, it's to show that it is safe enough to continue studying. A small group, typically 20 to 80 participants, is enrolled. In most therapeutic areas, these are healthy volunteers, people with no financial stake in the drug's success, who are compensated for their time. The exception is oncology. Cancer drugs are often too toxic for healthy people, so patients with the target disease are enrolled instead, and they may actually benefit. Phase 1 studies are conducted in specialized clinical pharmacology units, facilities equipped to monitor participants around the clock with frequent blood draws, ECGs, and vital sign measurements. 5 critical questions must be answered by the end of Phase 1. Is it safe? Is it tolerable? How does the body absorb and eliminate it? What biological effect does it produce? And what dose should be carried forward into Phase 2? The defining feature of Phase 1 is dose escalation, starting at a low, safe dose calculated from preclinical data and carefully moving upward, cohort by cohort. The traditional method is called the 3+3 design. Enroll 3 patients at a dose level. If none of them experience a dose-limiting toxicity, a predefined unacceptable adverse event, you escalate to the next dose. If 1 of 3 experiences a DLT, you expand the cohort to 6. If 2 or more experience DLTs, you stop. You have exceeded the maximum tolerated dose. The 3+3 is simple and well understood, but it has limitations. It can be overly conservative and may require many cohorts to find the right dose. Modern alternatives, the MTPI and Bowen designs, use Bayesian statistical models to continuously update decisions based on all accumulated safety data. They are smarter, more efficient, and increasingly preferred by regulators. Most Phase 1 programs also distinguish between single ascending dose studies, where each participant receives 1 dose, and multiple ascending dose studies, where repeated dosing is given to understand how the drug accumulates over time. SAD typically runs first, followed by MAD. Phase 1 introduces 4 terms that will follow you throughout your entire career in clinical research. Let's define each one clearly. Pharmacokinetics, PK, describes what the body does to the drug. How quickly is it absorbed into the bloodstream? Where does it distribute? Does it penetrate the brain? How is it broken down, typically by liver enzymes? And how is it excreted, through urine, feces, or breath? Key PK parameters include Cmax, the peak concentration in blood, Tmax, the time to reach that peak, AUC, the area under the concentration time curve, which reflects total exposure, and half-life, which tells you how long the drug remains in the body. Pharmacodynamics, PD, describes what the drug does to the body. This might be measurable inhibition of a target enzyme, reduction of a biomarker, or change in a physiological measurement. The PK/PD relationship, how exposure relates to effect, is used to design the phase 2 dose. The DLT, dose-limiting toxicity, is a pre-specified adverse event so severe that it signals the dose is too high. DLT criteria are defined in the protocol before the first patient is dosed. The MTD, maximum tolerated dose, is the highest dose at which an acceptable proportion of DLTs are seen. It is typically the dose just below where DLTs become unacceptable. And the RP2D, recommended phase 2 dose, is what gets carried forward. In some cases, it equals the MTD. But in targeted therapies, where maximum dose does not always equal maximum effect, the RP2D may be a lower, biologically active dose.